together, and, and at the end of the day, and we need to face the reality that HD is a global problem, and that implies that it does require a global response. So what I'm going to talk about is, first of all, we'll give you a couple of basics on Huntington's disease, and then discuss with you how Huntington's disease presents clinically and what the natural history of Huntington's disease is. And I will describe to you various stages of Huntington's disease, and then close with discussing very briefly how frequent HD is, because I think that's one of the interests, sure enough, for our first meeting in the Arab countries, that we get a better handle as to the frequency of the disorder in the various Arab countries. So, I'm going to start with the basics of Huntington's. And I'm showing this picture to you, although it is a picture that is hard to watch because it reflects the reality of HD. The reality is here a lady from Venezuela within 13 years reduced to somebody who is entirely dependent on others for her existence, and reduced from proud person to a person who is emaciated and has reduced abilities. And I think that's important to keep in mind, that everybody who tries to help HD families and to interact with HD families is very aware. Huntington's disease here is the person who gave the name to the condition. And you can see he's very young, and he was very young, because that's a picture taken from the time when he published his one and only paper on chorea right after leaving medical school. He identified the fact that Huntington's disease is a genetic disorder, which implies that everybody, as we heard in the video at the very start of the meeting, that everybody who is a descendant from an HD-affected person has a 50% risk to inherit the Huntington disease expansion mutation, or in other words, he or she is inheriting 50% future, if you like. And therefore, Huntington's disease is very much a disease of families. And it affects individuals typically in midlife, around 40 years of age. It does imply, as we have seen in the pictures before, and as we have heard from the video, long-term care. Young people are aware, like we heard in the video in the beginning, young people, descendants, sons and daughters are aware that they may develop Huntington's disease, and obviously parents do worry about the future of their children. So the entire family is affected by the disorder. Now, how does Huntington's disease typically present? What are the key clinical features? And the first thing that I would like to draw your attention to is that the clinical presentation of Huntington's disease is variable. It's not the same all the time, despite the fact that Huntington's disease is a disorder that is caused by a single gene, it's a monogenetic disorder. However, as we will hear a little bit later and stress throughout the talk, it has an unusual mutation or genetic alteration at its root cause, an expansion of a triplet of CAGs in the first exon that's depicted here of the Huntington's disease gene. And that is important because there is a close correlation between the number of CAG repeats and the age at which you see the clinical signs and symptoms characteristic for Huntington's disease. The threshold overall is 36. That's a number to remember. And when you may develop the clinical features of Huntington's disease, above 40 CAG repeats, 40 and more, however, it's fully penetrant, and everybody who lives the expected span of life will display signs and symptoms of Huntington's. So here we'll see a lady walking in a mid stage of Huntington's disease, and you do see the movements of the hands and the arms and the unusual way of walking that is reminiscent of dance. Here you see a person in an advanced stage of Huntington's disease, where now the involuntary movements have spread to involve the shoulders and the thighs, and you can easily see what a problem that is for the affected person as well as for the caregivers to provide proper care. Here you see a young gentleman who looks quite different. What you see here is that this gentleman is walking in a stiff way, and he holds his left hand in an unusual way. It's dystonia. And here he is supposed to do an alternating movements, and what he does is a repetition of these movements. We see this again, and please note how the left-hand side looks like. That's referred to as dystonia. Here you see an even younger person affected with Huntington's disease, and here you see very clearly the unusual posture of the hands and the inability to sit up. So that's a severe dystonic presentation. And that's typical if you have long CAG repeats, and is quite prominent, dystonia and Parkinsonism, if you have a long CAG repeat. Very nice study by Nando Squitieri detailed the phenotype of people with very long CAG repeat expansions who present early in life. So the very first conclusion, that's an important one, is that the way a person affected and carrying an expansion mutation in the Huntington gene presents clinically does depend on the size of the CAG expansion mutation. If you have normal, quote unquote, and the size is 43 CAG repeat and or like, you're presenting with chorea, which I showed in the first videos. If you're having more expansion, it looks quite different, and Parkinsonism and dystonia are in the forefront. So that emphasizes that the starting point of everything is the dynamic Huntington mutation that was discovered in 1993, so 28 years ago. And what was identified early on is between generations, if it's inherited from the mother, the offspring, the children, have very similar CAG repeat sizes. That's not true if it's inherited from the father, as you can see here, and there is a marked increase in CAG repeat sizes. So not only that you find this between generations, you find this in everybody who is carrying the HD expansion mutation gene. The CAG repeat numbers in the body are unstable, and they do increase during life. That's referred to as somatic instability, and you can see that's pretty dramatic. Here you see on B on the right hand side a person with 51 CAG repeats, and you see and here the little dots, and it can increase up to 700 CAG repeats in certain parts of the body. Here, the part of the brain striatum that is particularly affected. You see it also in the cerebral cortex, and it is so another part of the brain important for cognition and other functions. Interesting enough, these expansions appear to occur preferentially in cells that do no longer divide, that is in the nerve cells, and that implies that DNA replication, as for cell division, is not required. However, it implies DNA repair, because DNA can get damaged and it is used. And that is supported by big genome-wide association studies where we identified genetic modifiers here, a Manhattan plot where you see a couple of genetic modifiers and identified on various chromosomes, here a very prominent one on chromosome 15, that all belongs to the category of DNA repair enzymes. Here's an incomplete list of the ones that have been identified and that are in. So what we start to realize is that this fact, somatic instability, and still currently a hypothesis, appears to drive both the onset of the disease as well as the progression of the disease, and may explain the fact that there are dynamic DNA alterations, that there is late onset and a slow progression of Huntington. Let us look now a little bit more carefully in the natural history of Huntington's disease and the clinical features that emerge. Some of them I showed to you already in the videos. Here is this taken from a very nice paper by Francis Walker, published in the Lancet the other day, and gives you a picture of what is happening in Huntington's disease. And the first thing that we need to realize is that the life cycle of Huntington's disease is pretty long. It's a protracted course where you have a long period where you have no obvious symptoms and signs, but then there starts to be a period where you develop diagnostic symptoms and signs. Motor signs is the currently used criterion in most centers that tells you that the clinical stage of Huntington's disease did start. And if we look at this in more detail, this is the period where the clinical diagnosis of symptoms and signs are made, despite the fact that you obviously have the condition, that is the genetic change, from the moment of conception onwards in your body. So let's have a first look at the clinically silent period or asymptomatic period. Sarah Tabrizi with her co-workers looked at this in great detail in people who are very far away from the onset of Huntington's disease. It's called HD-YAS study. And she did a very detailed examination of imaging. Here is an example of imaging of wet biomarkers, things that you can measure in CSF as well as in blood, and very detailed and cognitive exploration. And what you see here, and that's quite interesting, is that there is no clear difference to normal people in all of these features, with the potential exception of one marker that you can determine in blood and in the cerebrospinal fluid, called NFL. So if we put this together in a scheme, you can see, sorry for that, you can see that about 20 years prior to the predicted onset of clinical symptoms and signs, you cannot distinguish clear-cut features. However, there's a long period of about 20 years, a little bit less than 20 years, where you have subtle symptoms and signs emerging. So let's look at this. What are the emerging clinical symptoms and signs in these stages of Huntington's disease that we refer to as prodromal? You don't have the diagnostic and motor signs that are characteristic for the manifest stage of Huntington's disease. First of all, it's important, although I have stressed and showed you in the videos motor signs, that this disorder is a neuropsychiatric disorder. So you do have personality changes, cognitive deficits, along with a movement disorder. And all this is reflecting the fact that you have a disruption of circuits that connect the prefrontal cortex, the basal ganglia, the thalamus, and back to the cortex. So this entire circuit is altered, and therefore you have a trias of affected signs and symptoms, thinking changes, behavioral changes, as well as motor changes. But it's important to realize that the changes are not restricted to what you have in the brain. You have the Huntington disease gene in its normal and in its altered mutant form in the body. And as a result, you do see, if you look closely enough, alterations in muscle, in heart, in the pancreas, and in other parts of the body. So if you broaden it up somewhat, you can say that we have, in fact, 4 domains that are altered in Huntington's disease: the movement disorder, the behavioral alterations that Alsbeta will discuss about, and we will hear about the movement disorders in detail as well, cognitive signs and symptoms, as well as changes in metabolism that results at the end of the day in people affected with Huntington's disease typically losing a lot of weight. Many of these alterations are caused are the consequence of the brain shrinking. You see here to the left-hand side a normal brain, to the right-hand side a brain of a person affected by Huntington's disease, and you can very clearly see that this part of the brain, the so-called basal ganglia, are massively altered. And you see, but in addition, however, that the white matters are far smaller in Huntington's disease patients compared to controls, and you see that the thickness of the cerebral cortex is markedly reduced in Huntington's compared to controls here. So it's interesting again to relate it back to what we have talked about earlier, that the extent of striatal atrophy that serves as a staging system for the extent of damage to the nervous system is again related to the CAD repeat expansion. So onset and progression are driven by the exposure to the mutant Huntington gene products, and you can bring this together in a formula that allows you to then have one single x-axis that is the product of age and axis CAG repeat expansion, and the various symptoms and signs, and as a in a reference as a percentage. And you can see then that depending on what you're looking at, you have various times of onset and emergence of symptoms and signs, some earlier, some later, and in a fairly characteristic fashion. And if you add red markers, things that you can examine in blood and in CSF, and for instance, neurofilaments, and Ed Wild did a terrific job exploring this, you can see that there are also clear alterations that you can measure and pick up both in the CSF as well as in the plasma. And if you put all this together, you can come up with a sequence of things. For instance, the first thing is the emergence of, unsurprisingly, the mutant Huntington elevated in the cerebrospinal fluid, then other markers like plasma, NFL, then changes in the brain that you can only pick up with imaging before it starts to show up in clinical symptoms and signs that you can pick up. So that gives you a sequence of events. Look at the rate of progression is also equally interesting, that the rate of progression is quite variable. Here you see 2 examples, fairly shallow, fairly steep. And so what determines that? That's an important question for us to understand. And again, it appears that a CAD repeat length appears to be critical here. You have faster progression if you have a long CAG repeat expansion. And if you look what is an important driver at the biological or molecular level, it's again a DNA repair protein, MSH3, that has been identified. And if you take in animals away MS3 totally, you will see that the expansion, somatic expansion that you see here and to the right-hand side, to the right hand of this red line, will disappear altogether. So that's again important, and repeating the same theme. So if you think about this, and in a summary, the disease symptoms and signs emerge. Very often people do notice that something is not quite right prior to anything overt being altered. You can then make the clinical diagnosis based on motor symptoms and signs, and that all this reflects the status of your nerve cells in your brain. With a decreasing health of nerve cells, and you are in a state of neuronal dysfunction that eventually will become irreversible with the loss of nerve cells. But it's quite important to realize that for Huntington's disease, you have a very long period extending over several decades, where you have neuronal dysfunction and where you can reverse things and where you can hope to have an impact with your treatments. So let's now finish with the stages of the Huntington's disease. And there's a new system that I would like to introduce to you, a new system that was developed by the HD Regulatory Science Consortium, a working party there, that wanted to align the staging system with other neurodegenerative disease, namely Alzheimer's disease. So you have Huntington's disease as a genetic biological entity, then biomarkers of pathogenesis, and following up, and then clinical symptoms and signs, and then the onset of disability or functional change as the sequence. That results in these 4 stages, and that's a proposal by this working party, where you add for each stage one particular determinant or threshold when it emerges, and so that you end up with Huntington's disease just defined by the presence of the genetic alteration, then biomarker alterations suggestive of biological damage, then first clinical symptoms and signs emerging, and then the functional alterations of Huntington's disease that imply that you're in need of assistance, and where the traditional clinical staging system based on total functional capacity is based. So the third stage here in this new integrated staging system can be further differentiated by the use of a rating scale called total functional capacity rating scale. Now, last point that I would like to make is to briefly discuss with you how frequent HD is, and the conventional wisdom that we have in our textbooks is that you have in the Western world a frequency of 10 to 14 individuals per 100,000 inhabitants. If you want to read up all the evidence for this, there's a very nice systematic review by Sir Michael Rowlands on this topic. What is interesting is that over the last decades, it appears that the HD prevalence is on the rise, at least in North America, Australia, and Western Europe. And if you look, and there are very few studies on that, and how frequently you do observe the Huntington expansion mutation, you see something quite interesting. And this is a nice, although preliminary study from our colleague Ahmad Aziz, now working in Bonn, where he looked at the prevalence of carriers of intermediate and pathological polyglutamine alleles, and he found that's depicted here in red that a surprisingly high number of people are in a pathological range that is above 36, and you can see here the number of people studied overall, and 19 out of those 12,000 people studied had crossed the threshold of 36 CAG repeats. However, did not quite reach, or not many of them reached, the full penetrance range, that is 40 CAG repeats. So the implication of this is that if you look genetically in a population-wide study, and what I just presented is not a truly population-wide study, then it may well be that the frequency of the Huntington disease mutation is one order of magnitude higher than the number of people who have clinical Huntington's disease. So Huntington's disease has a worldwide distribution. Michael Hayden studied that quite extensively with his group, and I'm showing one of the distribution studies based on the reported literature that he published. And it's quite interesting that it appears that there is a correlation between the average CAG repeat length in each population and the frequency of Huntington's disease overall, with quite some difference between Europe at this point in time, Asia, and countries like continents like Africa. So let me summarize. HD, and I think that's our motivation to work on this, and it is a devastating disease of families, and we would like to make a difference here. Huntington's disease is a monogenetic and autosomal dominant disorder. It's fully penetrant with the CAG expansion larger than 36 in the Huntington disease gene. The age of onset, the form of motor presentation, as I showed to you, chorea, dystonia, bradykinesia, the rate of progression, and the age of death. All, I didn't show you the data for the age of death, all appeared to be driven by the size of the CAG repeat expansion. And also the prevalence in a given region appears to be correlated with the average CAG repeats in the length in the Huntington disease gene in this particular population. So that's an overview, and we will have the pleasure to go deeper and dive deeper in the following presentation. Thank you very much for your attention.